Hello, we will present about inCCsight, a web-based software for Corpus Callosum DTI data processing, exploring, and visualization. This work was developed by Thais Caldeira, William Herrera, Aline Lapa, Professor Simone Appenzeller, and Professor Letícia Hitchner, all from Unicamp.
The Corpus Callosum, or CC for shorts, is the greatest white matter structure in the human brain and connects both hemispheres. Its properties and characteristics have been extensively studied and related to sex, laterality, intelligence, as well as conditions such as autism, epilepsy, psychiatric diseases such as depression and schizophrenia, alongside degenerative diseases like Alzheimer's and Parkinson's disease. Due to its highly organized fibers, analysis with diffusion tensor imaging, or DTI, has provided new relevant information about the corpus callosum. DTI is a type of magnetic resonance exam that collects information about how the water moves inside tissues. In the brain, this gives us information about how the fibers and paths are organized and is especially rich for corpus callosum analysis. Usually, studies of the CC in DTI follow a pipeline as shown. The diffusion data is acquired in an MR machine, followed by pre-processing and obtaining the tensorial image and its scalar maps. Then there is the segmentation, which means determining its limits, and parcellation, which is the division of the structure in different parts according with the cortical regions with which they are interconnected. Finally, all this information turns into numbers, tables, and statistics. We defend, however, that this pipeline is not quite robust, especially when working with large datasets. It requires knowledge about several softwares for each step, and often coding skills, making it inaccessible for a lot of researchers. Often, there is no quality control when using automatic segmentation and parcellation methods, making studies prone to errors and misinterpretations. There is low to non-repeatability and reproducibility due to the use of different programs and scripts, leading to unreliable studies. And finally, the final data is only looked at as numbers, but there's not a practical platform to check visually for abnormalities in the DTI. For all these flaws, we propose to concentrate at least part of this pipeline in one single software, to simplify the life of researchers and empower them to make reliable, reproducible, and meaningful studies. This is inCCsight. It is a web-based software that allows DTI processing, automatic segmentation and parcellation of the corpus callosum, quality check, and provides a dashboard for quantitative and qualitative data exploration with several interactive graphs. It is open source and available for Linux, Windows, and Mac. In the quality evaluation panel, the user can check those segmentations that were considered a potential error. There, we can check the reason the program detected it, check which are indeed wrong, and select them for removal so they don't impact the results. In case of wrongful removal, they can also be restored. If the researcher wants to check a specific subject, he can look for it in the subject list, which will open a panel with individual data. There are tables with segmentation and parcellation metrics, and there's a scalar maps visualization, where we can select the segmentation method applied, as well as the scalar map. The processed data can also be exported in a tabled manner. The user can select data from the segmentation and parcellation method he wants to export, which data to include, if he wants to add the standard deviation, as well as if he'd like individual or grouped data. In the dashboard, the user can find tabular data, where he can check results of different methods applied and organize the table in an individual or grouped view. We also present several interactive graphs. The scatter plot allows crossing information about different scalars and check trends in data. We also provide auxiliary graphs. The box plots show quartile information and reveal outliers. The scatter plot matrix allows us to check how all scholars are related with each other. The bubble plots and the midline plot are different visualizations for data sampled along the Corpus Callosum body and review group difference by section. All graphs can be manipulated interactively to help the researcher to find abnormalities and test hypotheses, and can be saved with publication quality. In conclusion, we believe all these functionalities can help researchers all over the world to perform Corpus Callosum studies in DTI in a practical, reliable, reproducible, and repeatable fashion, without the need to code or use several programs. We also believe that our dashboard allows data exploration and may help researchers discover new and meaningful information about this unique structure. Our tool is available on GitHub and Docker. Please do check out and collaborate with us. Thank you for watching.